Hello, hi, good evening, everyone. Am I audible and visible? Okay, thank you. Good evening. You were not there in the last class. Okay, okay, no problem. We have discussed the week 3 or week 4 questions in the last class video, so you can check it out on the YouTube. Have you seen the video? Okay. You understood or you have some doubt from that class? Okay, if you have any doubt, then you can ask it. Okay, then should we wait for some time or we can start? I don't think so. Others will join. Okay, have you solved the assignment of this week 5? I think that due date for week 5, okay, week 4 assignment, the due date is over. Okay, no problem. We will solve this. So, hello everyone, good evening. My name is Kajal Kachawa. For a short introduction of your TA, I will be your teaching assistant for this particular course of host pathogen interaction, the duration for which is from January to April 2025. So, today we are about to solve the week 4 assignment, and the video lectures for week 1 to 4 is already released on the YouTube. So, if you have not watched, you can check it out there. So I take every Saturday session from 3 to 5 p.m. in the evening, so you can join this. And till now on the NPTEL portal, the content and assignment for week 6 is released, so you can check it out there. And today we will be solving the week 4 assignment that is about the basics of cytokine and other immune mediators. So today we are going to learn about cytokines, which are special proteins that help cells communicate with each other, especially immune cells. Like these are a group of proteins which facilitate cell to cell communication between immune cells, and they also facilitate the interaction for development and the immune response. So, for a basic understanding, you can imagine like cytokines as messengers that send important signals between cells to regulate immunity, inflammation, and healing. Without cytokines, our immune system would not work properly, as our body would not be able to fight infections or heal wounds. So, basically, what are cytokines? These are a type of proteins that helps cells talk to each other. So they are important for the immune response because they help in activation of immune cells to fight against infections, and they control the inflammation to prevent excessive damage to the cells. And they also help cells to grow and repair tissues. And these cytokines are made by immune cells such as T cells. So these cytokines are made by T cells, macrophages, and dendritic cells, and also by the other body's immune cells. So, like, there are three mechanisms or the three ways by which these immune cells can work in the field of cytokines. Like, they can release the cytokines, and these cytokines will reach to other nearby cells, and there will be a downstream signal pathway. So, like, there are three types, three mechanisms of action. First one is the this autocrine action. It is the cell signaling. As we can see from the image, in the autocrine mode, the cells that make the cytokine also respond to its own signal. Like the cell is making cytokine and it is responding to its own signal. So, like for example, the T cells which release interleukin 2, it is an example of this. Like this T cell which releasing interleukin 2, this act by all three type of this modes like autocrine, paracrine, and endocrine. So in autocrine, what is happening? It is self signaling. And in paracrine, what is happening here? It is the neighbor signaling. Neighbor signaling means in the paracrine mode, cytokines act on nearby cells. They are released by one cell and they affect the nearby cells to help them respond to infection or injury. And the example of this is like macrophages detect bacteria and release cytokines, activate nearby WBC cells for helping them to fight with the infection. Like this macrophages are the examples of this type of signaling, that is paracrine, which is neighbor signaling. And the third one is endocrine action. Endocrine is like long distance signaling. So, what is happening here in the case of endocrine? Cytokines travel through the bloodstream to reach and act on the distant cell in the body. Like one cell is producing the cytokine, it will be traveling through some bloodstream and it will be reaching to some distantly located cell. Like the example of this is during an infection, the immune system release TNF alpha. That is a factor, that is tumor necrosis factor alpha, into the bloodstream. And this signals the liver and the brain to activate the fever response to help fighting the infection. So this is an example of endocrine. So, what we have learned from this first basic slide, there are three modes of the cytokine action. Like first one is autocrine, which is self signaling. Second one is paracrine action, that is the neighbor signaling. And the third one is the endocrine action, that is the long distance signaling. And the example of this autocrine is the T cells which release interleukin 2, and the example of paracrine is the macrophages which release cytokines to activate the nearby WBC. And the example of this endocrine is TNF alpha factor, which is released through the bloodstream for activating the fever response to fight from the infection. So these are the most common type of cytokine effects, and the T cell releasing interleukin 2 which can act by all these three type of modes. So I hope this basic types of cytokine release is clear to all of you. Please confirm. Okay. Okay. There are some properties of the cytokines. Like the cytokines are small regulatory proteins or glycoproteins that act as messengers in the immune system. They help immune cells communicate and coordinate towards the infection, injury, and other the threat to the body. And these molecules play a crucial role in regulating the intensity and the duration of the immune response, like how intense the immune response should be and for what duration the response should be there. These cytokines help in regulating that response, which ensures the body reacts appropriately to different kind of threats to the body, like different kind of infection or an injury or anything abnormal to the body. Cytokines respond to that. And these cytokines are characterized by several important properties, like these are low molecular weight glycoproteins or regulatory proteins. These are the why these are low molecular weight, because these cytokines are small molecules that can easily diffuse through the tissue or interact with the target cell. So that's why they are low molecular weight regulatory proteins. And this property helps them to act quickly and efficiently in the immune response. Like they can be taken up by nearby cells very easily because as they are low molecular weight, they can travel through the bloodstream because of their low molecular weight. If they are high molecular weight, then there will be problem. And the second property is that they are secreted by WBC and other cells. Like cytokines are mainly produced by the immune cells like T cells, macrophages, and dendritic cells. So if the question is which immune cells are basically produced from the like most common cells, which are most common immune cells which are producing cytokines, then those are T cells, macrophages, and dendritic cells. But there are other cells also, like fibroblast and endothelial cells, that secrete the cytokine in response to any infection or the injury. And the cytokines are like short lived, generally ranging from few hours to few days. Like they will be not for the like lifetime, that once a cytokine is produced in response to any type of infection or injury, and that will not last for like lifelong. They have a short lifespan of from few hours to few days, because these are not stored in the body. They are produced only when needed and they act for the short duration. Their action typically lasts for like few hours to few days, and this prevent excessive immune activation, which could lead to the chronic inflammation or tissue damage. If it is produced and stored in body for a very long time, then that can cause the irrelevant immune activation. Like whenever not needed, then also immune system will be activated, and that will cause the inflammation or some fever in our body as response to the cytokines. So these are short lived. Because of this property, they are short lived. And this cytokines can regulate the intensity and the duration of the immune response. How this is happening, like the cytokines can control how strong an immune response will be and when an immune response should stop, for what duration the immune response should be there. Like if an immune response is too weak, the body cannot fight from the infection. If the immune response is like too strong, then it can lead to autoimmune disease or a chronic inflammation or prolonged fever, which is not required for the body. So there should be a certain required intensity of the cytokine action and a certain required duration for how long it should be there. So these can regulate how the immune response will be and for what duration it will be. So these are the properties by which the cytokines are facilitating an immune response, and the cytokines exhibit its effect by stimulating the immune cell activation, by promoting inflammation to fight against infection, and by regulating tissue repair and healing, and by suppressing excessive immune response when required. So there are some other actions of the cytokines, which are till here, is this clear? What are different ways of cytokine action and what are the properties of cytokine action? And what is the relevance behind each and every property of the cytokine? Is this clear till here? Okay. And are you aware of these type of cytokine actions like what is pleiotropy, what is synergy, what is redundancy? Do you want to comment on this? What is your understanding about these terms? Mm-hmm. 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 Yeah, almost correct. No issue with this. Okay, I'll explain this in more detail. Like this pleiotropy is, in short, you can say this one cytokine, many effects. Like one cytokine and many effects. So, like what is happening here? A single cytokine affects multiple type of cells, and each cell responds differently. For example, if like interleukin 4 is released from this particular cell, that is the activated T helper cell, this interleukin 4 is released. And if interleukin 4 will signal to a B cell, then it will start like this activation, proliferation, and differentiation. Like it will start growing and maturation. And if this interleukin 4 is received by an thymocyte, then it will undergo proliferation. That will, like that, it will trigger the cell division, the proliferation. And if this interleukin 4 is received by a mast cell, then it will cause the proliferation. So there is difference. Like if B cell is receiving this interleukin 4, it will undergo activation, proliferation, and differentiation. If thymocyte and mast cells are receiving this interleukin 4, they will undergo proliferation. Even though the molecule is same, cytokine is same, that is the interleukin 4, but it have different effect on different type of cells. So like one cytokine can trigger different reactions depending on which cell it interact with. And this will help the immune system to do multiple signaling at a time. Like for triggering and B cell, there will not be a separate interleukin for the response for taking out the response from a thymocyte. There will not be a separate interleukin. A single interleukin can interact with different type of cells, and depending on the receiver cell type, the function will be different. Like one cytokine, many effects. So this is pleiotropy in general. And what is this redundancy? It is like different type of cytokines, but same job, same effect. Like there are, you can see there are three type of cytokines: interleukin 2, interleukin 4, and interleukin 5. Like in redundancy, there are three different type of cytokines. These are all secreted by this activated T helper cells, and these are received by this B cell only. And the effect of or the action from this all three type of cytokines is the proliferation. B cell will get proliferated, whatever is a cytokine, whether it's a interleukin 2, interleukin 4, or it's interleukin 5. So this is the example of redundancy. That is different cytokines, same job. And the third one is synergy. Synergy is like, it is something like teamwork, synergy, synergistic effect. Like synergistic means working together, like a teamwork. So this is synergy. So what happens here? Different cytokines work together to get a stronger effect than they could produce alone. Like in synergy, two different cytokines, that is interleukin 4 and interleukin 5, both are working together and they are generating a more stronger response for the B cells, that is inducing the class switching to IgG. This you will study in detail when you will be studying about the types of antibody production, antibody class switching in later units. There you will study in detail the class switching. So here the response of both interleukin 4 and interleukin 5 together, when they both are received together by the B cell, then the response is they induce class switching. So this is synergy. So in general, what happens here in the case of pleiotropy, what is happening? One cytokine, many effects. Like one cytokine, many effect from the different cells. And in redundancy, what is happening here? Different type of cytokines, but same job, same effect. In synergy, yeah, like you can write down these short terms along with you are listening so that it will be easier for you to learn. In synergy, what is happening here? It is a synergistic effect. That is the teamwork type of thing here, synergistic, working together. So if two type of cytokines, two interleukins are working together, they will push this B cells to switch the class to IgG, that is antibody class switching. So these are three type of cytokine action. Is this better clear now? Okay. And there are some other type of the cytokine action. These were like some good one, like some positive effects or positive function. There are some other functions also of the cytokine action, that is antagonism and this cascade induction. What are these? Antagonism and cascade induction. It is blocking that other cytokine, inhibitory effect, negative effect. And what is this cascade induction? What is cascade? Hmm. This one cytokine is inducing a chain of reaction for the downstream signaling. So, what happens in antagonism is like not all the cytokines will give a positive effect for the cell to function. There will be some inhibitory action or some like anti effect of a cytokine. So, in antagonism, what happens here? One cytokine block the function of other cytokine. Like one cytokine is blocking other. Like what was happening here? This activated TH cell was releasing interleukin 4 for the B cells, but then interferon gamma came in between and it blocked the effect of this interleukin 4. So this interleukin 4 will no longer be received by this B cell. So this interferon gamma is working as antagonism. So what will happen here? This will block the class switching of IgE induced by interleukin 4. Like in this slide, what we have seen, this interleukin 4 is responsible for inducing class switching to IgE in B cells. And when it receives this interferon gamma along with this interleukin 4, this interferon gamma will cancel out the effect of interleukin 4 and it will block the class switching induced by the interleukin 4. That is, this B cell will no longer switch the class. So sometimes this is also required, otherwise, with the response to a minute amount of this particular cytokine, B cells will switch the class and it will start producing different type of antibody. But there should be a mechanism like how to stop this particular reaction of class switching. So there is a release of interferon gamma that will instantly block this effect of interleukin 4 and it will block the class switching. So this is the antagonism, like blocking the effect of other cytokine. There is one more action of cytokine, that is cascade induction. Cascade is like a chain reaction. So one cytokine will trigger a chain reaction. So what is happening here in this case, like as you can see in this example, so in this example, you can see that in the case of cascade reaction, what is happening? T helper cells, that is the activated TH cell, it release interferon gamma, which activates the macrophages. And this activated macrophage will then release the interleukin 12, which will activate even more T helper cells. And these T helper cells will again release more amount of interferon gamma, TNF, interleukin 2, and other cytokines. So, what was happening here? This activated T helper cell released interferon gamma. This interferon gamma activated this macrophage, and this macrophage again produced interleukin 12. And this interleukin 12 activated more number of T helper cells. And this more number of T helper cells released more amount of interferon gamma, TNF, interleukin 2, and many other cytokines. That is, it gave a more stronger cytokine action. Like this one cell was insufficient to produce this much type of cytokine. So it activated macrophages and it ultimately activated more number of cells, ultimately leading to production of more amount of cytokines, which gave a higher immune response. That is a more stronger immune response. So what is happening here? You understood this cascade induction. So what we have studied here, there are like different type of cytokine actions, how cytokines are working. Like they can work by the pleiotropy, that is one cytokine, many effects. They can work by redundancy. What is in the redundancy? Different cytokines, same effect. And what is here in synergy? Hmm. They will work together, yes. And what is here in the antagonism? Other cytokine. Yes. And in the cascade induction, yes, and ultimately leading to a more stronger immune response. So these were different type of cytokine actions. And now there is cytokine nomenclature and types, like what each type, each name of the cytokine represents to what. There is a nomenclature for cytokines. Like there are lymphokines. Lymphokines, from the name, we can see that these are secreted by lymphocytes. There are monokines, like these are secreted by the monocytes and macrophages. There are interleukins, like these leukins are secreted by the leukocytes. So this is the nomenclature. These are based, these cytokines are based on the type of cells which are secreting them. So what are like this lymphokines, this lymphocytes? Like there are T lymphocytes and B lymphocytes. If a cytokine is produced from B or T lymphocyte, its name will be like lymphokine. And this monocytes and macrophages will produce the monokines, and this leukocytes are the WBCs, white blood cells. These will produce the cytokines that will be known as the interleukins. And there are some other cytokines also called as like tumor necrosis factor, like TNF. These are named because they can kill tumor cells and regulate inflammation. Like there's necrosis effect, that is the killing effect for the tumor. So these are tumor necrosis factor, like TNF. And there are some chemokines. These are the small cytokines, low molecular weight cytokines that affect chemotaxis and other aspects of leukocyte behavior. Like these small cytokines can guide the immune cells to the right place, and they are providing signals to the cells. Chemotaxis is like providing some chemical signals to the cells that where the immune cells should move. Like body don't know where the injury is, where the inflammation is. There are these particular chemokines released. These chemokines will take other immune cells to the site of action. This process is called as chemotaxis. That is the movement of inducing the movement of other cells by the release of some chemical molecules. On these chemical molecules are the chemokine. Like those are the cytokines. So this is chemokine, and there are some cytokine types based on the source cells. That is the innate and adaptive immune cells, and based on their function, like the cytokine based on the function, there are like how these are divided. Like the cytokines can be divided on the way of their development of cellular or humoral immune response. Like the cytokines can help in activating the immune cells, and they also guide the E cell and B cell to recognize and respond to the pathogens like bacteria and viruses. And their function is to induction of the inflammatory response. Like inflammation is the body's first defense against injury. Like if our body is injured, then the first response will be inflammation. Just a minute. So, what we were talking about, the induction of the inflammatory response. So, this inflammation is the body's first defense response against any injury or the infection. So, like the cytokines like interleukins and TNF alpha that trigger inflammation by making the blood vessels leaky and allowing immune cells to reach the site of the infection faster. So these cytokine help in the induction of inflammatory response, and other their function is regulation of hematopoiesis, that is the blood cell formation. The immune cells needs a very high supply of fresh immune cells. Like the cytokines can help in this process, like the generation of new type of blood cells. And they can also control the cellular proliferation and differentiation. Like the cytokines can decide which immune cells should multiply and what specialized role should they have. Like it can help in the proliferation and differentiation into specialized cells. Like these can decide which cells will become the memory cells to provide the infection in the future, and which cells will produce antibody, which cell will go where. So these cytokines can control the differentiation of cells. The cytokines can also heal the wound healing like this. For example, the TGF beta. that is transforming growth factor beta, can help in repairing the damaged tissue after an infection or an injury. So this is the again function of cytokine only. So there are different function of cytokines like development of cellular and humoral immune response, induction of the inflammatory response, regulation of hematopoiesis, that is the blood cell formation, and the control of cellular proliferation and differentiation, that is the which cells will be formed and how those cells will be divided, and the wound healing. There are some like pathogens and their cytokine in response to what. So you can check it out from this table. So till here is this clear? Okay. So there are some this innate immunity cytokines and some adaptive immunity cytokines. So let's see what are these. Like this, some cytokines are responsible for providing the innate immune response. Like for example, this interleukin 1, which is produced by monocytes, macrophages, and like other immune cells. It is one of the main cytokine responsible for fever and the inflammation and the induction of acute phase proteins in the liver. So this is like this here. This is the function of this interleukin 1. And there are this tumor necrosis factor alpha, that is TNF alpha, which is released by macrophages, monocytes, neutrophils, and other activated T cells and natural killer cells. So these are released by these cells, and this TNF alpha can help with the inflammation, but in large amount, it can cause cell death. Like if more amount of TNF alpha is released, then it can cause the cell death also. And their other cytokine is interleukin 12, that is IL-12. It is released by macrophages and dendritic cells. And this help in the activation of this natural killer cells, and it influence the adaptive immunity by promoting TH1 subset. And there is like this interleukin 6, which is produced by macrophages and endothelial cells. It is also responsible for this adaptive influencing the adaptive immunity. And there is like interferon alpha, which is produced from the macrophages and dendritic cells. There is other cytokine, that is interferon beta, which is produced by macrophages and dendritic cells. So these are some of the cytokines which are responsible for innate immunity. And there are other cytokines like interleukin 12, interleukin 4, interleukin 5, and TGF beta and interferon gamma. These are responsible for the adaptive immunity. So, those were the types of cytokines of the innate and adaptive immunity. Next, there are some immune mediators. Just a minute. So, other than the cytokines, there are some immune mediators. These immune mediators are also like they have the function similar to the cytokines. So there is two main immune mediators, that is histamine and the serotonin. Have you heard these terms before, histamine and serotonin? Hmm. And serotonin, it is a neurotransmitter. In that context, I think you might have heard this. Okay, no problem. We will take this. So, like we generally think of serotonin as only a brain neurotransmitter, but it also have a key role in the immune system. It is found in like hematopoietic stem cells and is highly abundant in the platelets, the serotonin. And the serotonin regulates the inflammation and immunity by acting on serotonin receptors that are differentially expressed on immune cells. So, this is the structure of the serotonin. And the serotonin helps in regulation of inflammation and immune response by making sure that the right immune cell will go to the right place at the right time. So, this is the major key function of the serotonin, that it helps in the immune cells to go to the right place at the right time. And this serotonin also have role in the inflammation, like it act as a chemoattractant and it recruits the innate immune cells to the site of inflammation. And it also alters the production and release of the cytokines and cell activation and proliferation. And this immune cells, like in by inducing the this, here comes the role of like the histamine. The histamine this have this type of structure and serotonin have this type of structure. So this histamine have a major role in the allergy. This is called the allergic response mediator. So this histamine is released from which immune cells? There are some immune cells which produce some, that is the antibody IgE that get activated in the case of immune this allergic reaction. Which are the immune cells that produce this histamine? If you can recall the unit of the cells of immune system, then there I think you might have read about the mast cells, basophils. Those are the granular cells, like this granules containing cells, majorly the mast cells, and yes, granulocytes. So these cells contain granules, and these granules contain this type of this vasoactive amines like histamine and these allergy mediated compounds. So this histamine is produced by mast cell, basophil, and some other immune cells also. And it is known for its allergic reaction, but it also plays a key role in the inflammation and immune cell signaling. So this what this histamine will do, it will increase the blood vessel permeability and allow the immune cells to move into inflamed tissues faster. Like it will increase the blood vessel permeability so that the immune cells can walk out of that space and can reach to the site of inflammation. So it triggers the symptoms like swelling, redness, and itching. So this is the common symptoms we see in the case of allergy. Like if we get allergy, then some of our body parts may get swell up and redness and itching, like our nose, eyes, face, skin, or any space get swelled up and redness and itching. That is because of this release of histamine. So this histamine and serotonin are two type of immune mediators. So this serotonin is majorly involved in the case of inflammation, and this histamine is majorly plays a role in the allergic reaction. So see what is happening here in this case. One, an allergen that may be any like any pollen grain, any dust particle, or any like food allergen, like some people have allergy to nuts, milk, and other food items. So this allergen can be anything. So when an allergen enters the body, it interacts with the mast cell. This mast cell is the granulocyte, that is granule containing cell. And the mast cells will have this IgE antibody on its surface, and it will recognize this allergen, and it will trigger the degranulation of these granules which contain the histamine. So this allergen will come, it will bind to the antibody, that is IgE, situated on the cell surface of this mast cell, and because of this response, these granules will get degranulated, and the histamine will be released, and the inflammatory mediators will be released. So, what will happen once these inflammatory mediators or histamines are released? These histamines will act on different target cells, and it will cause different type of effects. Like when this histamine will come in the contact of smooth muscle cells, it will cause contraction, which can lead to symptoms like tightness in the airways. Like our nostrils and airways will get tightened, which happens in the case of asthma. Like this histamine will interact with the smooth muscle cells. Then it will cause contraction, which will lead to the symptoms like the airways tightening. And when this histamine will come in the contact of small blood vessels, it will cause them to expand, that is the vasodilation, and become more permeable, which allow the immune cells to move into the tissues. This is why swelling and redness occur at the site of inflammation, because the immune cells are moving into the tissue. So, and next, when this histamine will come in the contact of this mucus gland, what will happen? It will increase the mucus production, which is why this allergies often cause runny nose and watery eyes because of the interaction of histamine with this mucus gland. And when the histamine will come in contact with the blood platelets, it will help in blood clotting and wound healing, which plays a role like other than the allergies. And when the histamine come in the contact of the sensory nerve endings, it will stimulate itching and pain, which is why like this allergic reaction causes itchiness, itching in the eyes or on our skin because of this interaction with the sensory nerve endings. And when this histamine will come in the contact of eosinophil, it will attract the immune cells that are involved in fighting with this pathogen and parasites, and it will again regulate the allergic inflammation. So these are the different type of body cells which are interacting with the histamine in response to the allergy or inflammation, and each type of cell will have different type of action. So this is the action of histamines. Did you get this point? No confusion in this thing, what is happening? Okay. Okay, so now let's move to the week 4 assignment 4, the due date for which was 19th of Feb. So I hope you might have submitted this. We will take these questions. The first question is which of the following conditions is most likely to result from this regulated cytokine production? Like these are the 5 conditions. So, in which condition results from the dysregulation of cytokine production? So, would you like to answer for this? Autoimmune diseases? And what about other options? Hmm. No problem, I will explain this. But this is clear to you now that because of the dysregulation of cytokine production, autoimmune disease may occur. But you should also know about the other disease as well, like what is happening in those disease. So, in the case of Alzheimer disease, it is a neurodegenerative disorder which is linked to the protein misfolding and brain cell damage. So, like inflammation can play a role here, but cytokine dysregulation is not a cause of Alzheimer disease. In Alzheimer, protein misfolding and brain cell damage is happening. And in type 2 diabetes, what is happening? It is caused by the insulin resistance, which is linked to the like our dietary habits and obesity, and it may be genetic because of the genetics. And like there is no role of cytokines in this type 2 diabetes. And this osteoporosis is a bone density loss disease, like in this case. In the case of osteoporosis, bone density is lost, often due to hormonal changes or calcium deficiency or because of the aging. So there is a no role of cytokine in this case. And in the case of hemophilia, hemophilia is a genetic disorder where blood clotting factors are missing. So this have nothing to do with the cytokine dysregulation. So there are five different type of disease. Alzheimer disease is neurodegenerative disorder. Here protein misfolding and brain cell damage occurs. Type 2 diabetes is because of the insulin resistance, and osteoporosis is the loss in bone density, which is because of the hormonal changes, calcium deficiency, or because of the aging. And this hemophilia is a genetic disorder of blood clotting factors missing. So these are the you should also know about other type of disease as well. So the correct answer is autoimmune disorder. Here the dysregulation of cytokine production happens. Like we have studied, now if a cytokine is producing in more amount, so that will cause autoimmune disorder. So the second question is, what is the primary impact of vasoactive amines like serotonin and histamine in the immune system? We have just seen this. What is the correct answer for this? How this happening? Why not this inhibiting cytokine production or other options? Hmm. Hello, am I audible? Am I audible? I think there is some network issue. Yes, yes, you are audible, but I think there is some network issue. No problem, I will explain this. So, here the question is, what is the primary impact of vasoactive amines like serotonin and histamines in the immune system? So, like we have just seen that this cytokines and vasoactive amines work together in the immune response. But histamine and serotonin don't directly inhibit any type of cytokine production, but they actually help in the regulation of inflammation by promoting the immune signaling. So they both will not inhibit the cytokine production. And the second option is preventing allergy reaction. So no, histamines are responsible for causing the allergic reaction. They are not preventing, but they are triggering the allergic reaction. So this is also wrong option. The next option is enhancing T cell differentiation. So the T cell differentiation is mainly regulated by cytokines like interleukins, some of the interleukins like interleukin 2 and 4, but not because of the vasoactive amines. So here we are discussing the role of vasoactive amines like the serotonin and histamines. So they do not have any direct role in the T cell differentiation. Then the next option is regulating inflammation and allergy reaction. So yes, this is the correct function of this serotonin and histamine because the histamine triggers the inflammation by causing blood vessels to expand and become leaky and allowing the immune cells to reach to the site of infection. And the serotonin also plays a role in immune cell movement and cytokine release by the mechanism we have discussed that chemotaxis. So these both have a role in inflammation and allergic reaction. And stimulating antibody production, no antibodies produced by the B cells, which have no direct role of this serotonin and histamine in the antibody production. So the correct answer is regulating inflammation and allergy reaction. So the next question is which of the following is a non-immune function of cytokines such as interleukin 8 and MCP1? So what is the correct answer for this? Hmm. Like what are these interleukin 8 and MCP1? What are these cytokines? What is their role? Interleukin 8 is mainly involved in recruiting neutrophils to the site of infection, and this MCP1 is monocyte chemoattractant protein 1. This have a role in attracting the monocytes and macrophages to the inflammation tissues. So their both role are of the chemoattractant. And this interleukin 8 recruit the neutrophils to the site of infection. So here the question is which of the following is a non-immune function of cytokines? So the regulation of ovulation, follicular development, and steroidogenesis. So this is the correct answer because the cytokines like interleukin 8 and MCP1 have been found in the ovarian follicles and this play a role in the ovulation, follicle maturation, development, and the steroidogenesis, that is the production of steroid hormone. And this MCP1 protein, that is the this MCP1 protein is involved in the follicular rupture during the ovulation. And this interleukin 8 contributes to the angiogenesis, that is the formation of blood vessels in the ovarian tissue. So uh, have a direct role in the ovulation follicular development, but this is not related to the immunology. This is not related to the immune function of these two things. So here we need to opt out the option that is the non-immune function. And these both have a direct activation a role in the direct activation of T cells during pathogen recognition and the production of antibodies in response to bacterial infection. No, this is not their role. They don't produce any antibody. And the enhancement of antigen presentation by dendritic cells, whose function was to present the antigen to the dendritic cells. There was something other related to this. Hey, can you find this? Whose function was to present the antigen? See, you can see here in this table the interferon gamma. It activates the macrophages and increases the expression of MHC class 1 and class 2 molecules, and it increases the antigen presentation. So it was the function of interferon gamma. And I think one interleukin was also there. Like this interferon is mainly involved in this antigen presentation because they are increasing the MHC class 1 and class 2 expression. And I think one interleukin is also there. You can cross check it whether it's interleukin 2 or interleukin 12. One interleukin is there, but this particular is not involved like this. What is asked in question is interleukin 8 and MCP1. These are not involved in the antigen presentation by dendritic cells. And the regulation of circadian rhythms, which cytokines are involved in the regulation of circadian rhythm. Here it was written somewhere. It is not directly mentioned here, but you can see, check it out. Like I think tumor necrosis factor, some TNF alpha or beta, something is involved in this regulation of circadian rhythm. So basically, you need to remember the cytokines, different type of cytokines of both innate and adaptive immunity, and their role and their function. Because there are so many questions related to this. So this regulation of circadian rhythm is, I think this is controlled by some of the tumor necrosis factor, but not because of this interleukin and MCP1. So the correct answer for this, the non-immune function of cytokines like interleukin 8 and MCP1 is the regulation of ovulation, follicular development, and steroidogenesis. So the next question is, how has genetic engineering impacted cytokine therapy? So what they are asking here, the role of genetic engineering in cytokine therapy. Hmm. Hmm. So, cytokine therapy is basically used to treat various conditions, including cancer, autoimmune disease, and viral infection. And the production of cytokines for medical use has been because of this genetic engineering, majorly the recombinant unit technology. So, cytokine therapy has made it, this genetic engineering has made it feasible to produce essential proteins for therapeutic purpose. Like we can mass produce these therapeutic proteins, like we can produce an higher amount, higher quantities because by using this genetic engineering. And it has decreased the production of cytokine for clinical use. No, it have increased the production of cytokines. It has slowed the development of antiviral therapies. No, it have in fact increased the development of antiviral therapies. It has eliminated the need for interferons in treating viral infection. No, it have increased the need for interferon in treating viral infections. It has only affected cytokines unrelated to interferons. No, this is also wrong. So it is very direct that it has made it feasible to produce essential proteins for therapeutic purpose. Like there was one slide, the cytokine therapy was initiated because of the advancement in genetic engineering techniques such as cloning of cytokine genes and generation of soluble cytokine receptors and generation of monoclonal antibodies directed against cytokines and the cytokine receptors. So this is the role of cytokine based therapy. Next question is, what are some of the diverse applications of interferons in medical treatment? So what are the applications of interferon? Hmm. 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 Okay, correct. So in the treat. Hmm. Hmm. Okay, correct answer. So these interferons in medical treatments are used for the treatment of viral infections. So interferons like interferon alpha and interferon beta are used in antiviral therapies. For example, the interferon alpha is used in the treatment of hepatitis B and hepatitis C, and in other this viral infections like HPV, that is human papillomavirus, and herpes, these interferons can also be used to reduce the viral replication. So there are many examples of this viral infections where interferon therapies, interferons are used. And in case of cancer therapy, yes, there also interferons are used. These interferons can help in activating the immune cells like NK cells and cytotoxic T cells to attack the tumors present in the body. For example, the interferon alpha is used to treat the leukemia, melanoma, and the type of cancers. So yes, it can be used in the treatment of like in the case of cancer therapy for increasing insulin sensitivity. So no, there is no need for this increasing the sensitivity of insulin. And in the case of immunotherapy, yes, we are studying from so long time that immunotherapy and this interferons have a major role in the case of immunotherapy, which can increase the immune system, like which can increase the immune response to fight against the disease or the infection. And the next option is stimulating stem cell differentiation. No, this is not a function of the interferon. There are some different cytokines which can influence this stem cell differentiation, but interferon is not involved in this. Healing bone fracture. No, interferon do not have a role in any type of healing the bone fractures. And the next option is neurological conditions like multiple sclerosis. Yes, interferon beta is majorly used for the treatment of multiple sclerosis. And this multiple sclerosis is an autoimmune disease where the immune system attack the myelin sheath, that is the nerve insulation. If you remember the structure of the neuron, then there is a myelin sheath surrounding or the outer covering of the neuron that is for the nerve insulation. So this, in the case of multiple sclerosis, immune system attack this myelin sheath. So in this case, interferon beta can help in reducing the inflammation and slowing down the disease progression. Next option is for reducing neuroinflammation. So yes, in this case also interferon can be used. Interferon beta is majorly used in the case of for controlling the excess inflammation in the case of neurological disorders. So these highlighted options are the major applications of the interferon in medical treatments. See in this slide, hepatitis B and C along with the ribavirin and interferons can also be used in the case of hairy cell leukemia, that is a type of B cell leukemia. In chronic myelogenous leukemia, CML also we can use this interferon. In Kaposi sarcoma also we can use. In multiple sclerosis also we can use this interferon. And there are some other conditions as well, like follicular non-Hodgkin lymphoma, cutaneous T-cell lymphoma, multiple myeloma, hereditary immunodeficiency, chronic granulomatous disease, osteopetrosis, which is a life-threatening congenital disorder characterized by overgrowth of bone that results in blindness and even the deafness. So in these cases, the interferon can be used. These are the examples of the disease condition where interferon can be used. So the next question is, what is the role of GM-CSF in immune cell development? So what is this GM-CSF? Hmm. Yes. So GM-CSF is granulocyte macrophage colony stimulating factor, and the role of GM-CSF in immune cell development is to it causes the hematopoietic stem cells to differentiate into dendritic cells. So here you can find it the GM-CSF it stimulates the production of myeloid cells after bone marrow transplantation. That is the it differentiates the stem cells into the dendritic cells. So what happened in this case? This GM-CSF plays a key role in directing the hematopoietic stem cells to become dendritic cells, macrophages, and granulocytes. And these dendritic cells are essential for antigen presenting cells that can help in the activation of T cells and coordinate the immune response. So, this GM-CSF is majorly used in immunotherapy to increase the dendritic cell production and enhance the immune response against cancer and infection. So, this GM-CSF had a major role in differentiation of hematopoietic stem cells into dendritic cells. And what about other options? Is it involved in the production of antibodies? GM-CSF is not involved in the production of antibody. Antibody is produced by the B cells and it inhibits T cell proliferation. No, it will not inhibit the T cell proliferation. In fact, it will enhance the T cell activation by increasing the number of antigen presenting cells like the dendritic cells and macrophages. So it will not inhibit the T cell proliferation. It will enhance or increase the T cell activation. It acts as a hormones that regulate glucose level. No, don't have any role in the regulation of glucose levels. It is regulated by insulin and glucagon, not the GM-CSF. It promotes wound healing. No, there are other cytokines like some TGF beta and some other interleukins which can help in wound healing, but GM-CSF don't have any role in the wound healing. It is mainly involved in the immune cell activation, like that, this production of dendritic cells. So, the role of GM-CSF is to cause the hematopoietic stem cells to differentiate into dendritic cells and increase the number of antigen presenting cells. So, these are the cytokine based therapies. Here you can find it GM-CSF. The next question is which of the following are considered pro inflammatory cytokines? There are two type of cytokines. One is pro-inflammatory and other one is anti-inflammatory. From the name, you can understand this pro-inflammatory will promote the inflammation or the immune response, and the anti-inflammatory will suppress or inhibit the inflammation. So, which of these cytokines are pro-inflammatory? Hello, are you speaking something? Yes, yes, you are audible. Mhm. Can you repeat what you just said? Hello? Shweta, are you speaking something? Are you asking? I think there is some network issue, so you can write in the chat box. Hi, is your network stable now? Yes, yes, you are audible. You can write in the chat box if your network is not stable. No issue. Shweta, are you typing something or should we proceed? Can you hear me now? Shweta, am I audible? Shweta, can you hear me now? Yes, yes, you are audible. Can you hear me? Okay, no problem. Two, three questions are remaining. If it is stable now, then we can continue. O- Okay, you can like interrupt me or stop me anywhere if you lost out the voice of me so that I can repeat that again. Otherwise, we can start. Okay. So, what we were discussing about pro-inflammatory and anti-inflammatory cytokines. There are two types of cytokines. One is pro-inflammatory. From the name, we can see that this type of cytokines will be responsible for activating the inflammation or for increasing the inflammatory response. And other one is anti-inflammatory that will inhibit the inflammatory response or will suppress the inflammatory response. So in this particular question, they are asking the examples of pro-inflammatory cytokines. Which of these are pro-inflammatory cytokines? So from this table, I have taken this, that is interleukin 6, TNF alpha, interleukin 1 beta, MCP1, GM-CSF. These are the examples of this pro-inflammatory cytokines. I will quickly help you in revising where these type of cytokines are used. Like this interleukin 6, it increases the inflammation, fever, and acute phase protein production in the liver. From the lecture videos, you can cross check that. That is interleukin 6 plays a role in autoimmune disease like rheumatoid arthritis. So, this is a pro inflammatory cytokine. The next one is TNF alpha, that is tumor necrosis factor alpha. It is also a pro-inflammatory cytokine. It is one of the most important inflammatory cytokine because this cytokine causes fever. It promotes the immune cell recruitment and it plays a role in autoimmune disease like rheumatoid arthritis and Crohn disease. And the next one is interleukin 1 beta. It is also a pro-inflammatory cytokine. It also triggers the fever, inflammation, and white blood cell activation, that is WBC activation. It is also involved in the autoimmune disease and inflammatory response to the infection. Next one is this MCP1, that is monocyte chemoattractant protein 1. It attracts the monocytes and macrophages to the site of infection or the site of inflammation. So it also plays a role in chronic inflammatory disease. The next one is GM-CSF, that is granulocyte macrophage colony stimulating factor. It is also pro-inflammatory. We have just recently seen this, that it stimulates the differentiation of immune cells, that is macrophage dendritic cells and neutrophils, and it promotes the inflammation and immune response. So these highlighted one are the examples of pro-inflammatory cytokines, and the remaining one, that is this TGF beta, that is transforming growth factor beta. It is an anti-inflammatory cytokine. It helps in it suppress the inflammation and it promotes the tissue repair. So it helps in controlling the immune response and preventing the excessive damage to the tissues. So it is an anti-inflammatory cytokine. The other one is interleukin 4. It is also an anti-inflammatory cytokine. This promotes the T helper cell response and it also helps in the allergic reaction and antibody production. So it is also anti-inflammatory. The other one is interleukin 10. It is also an anti-inflammatory cytokine. It also suppresses the immune response to prevent the excessive inflammation in the body. So it helps in the regulation of immune balance between the anti-inflammatory therapies. So it is also anti-inflammatory cytokines. So I have here quickly summarized the role of these different type of cytokines that you can remember from this table of pro-inflammatory cytokines and anti-inflammatory cytokines, where their name and their function is written. So you can check it out from the video lecture as well. These are the again pro-inflammatory cytokines. The next question is, what is the primary application of granulocyte colony stimulating factor, that is GCSF therapy? So what is the primary application of this? Yes, we. Hmm. So their major application is to enhance the neutrophil production. So from here we can see that GCSF it has stimulates the production of neutrophils, reduction of infection in cancer patients treated with chemotherapy and AIDS patients. So its role is to stimulate the production of neutrophils. So the application of GCSF therapy is to enhance the neutrophil production. And the next question is which of the following best describes the function of type 1 interferon? It's type 1. Yes. Yes. So the function of type 1 interferon is they interfere with viral replication and exhibit anti-proliferative activity. So how they do this? Like they block the viral replication by degrading viral RNA and by stopping the protein synthesis machinery in infected cells. So no protein synthesis machinery, no RNA will be produced, and this viral replication will not be there, it will not be active. So they block this viral replication machinery. And they also activate the NK cells, that is natural killer cells, to destroy the virus infected cells. And this type 1 interferon also increase the MHC class 1 expression and make the infected cells more visible to the CD8 cells. And they also have the anti-proliferative effects, means they can slow down the tumor growth and can be used in the cancer therapy. So the function of type 1 interferon is to interfere with the viral replication and exhibit anti-proliferative activity. And the last question is which interferon is primarily used in the treatment of multiple sclerosis? We have seen this beta 1A. Yes. So where are these other interferons used? Yes. Say this here if we take a quick summary of this interferon, but somewhere here in the table. Diseases were there no, in some slide. Here, interferons and treatment. So here for the hepatitis, yes. And this red one is for interferon beta. Yes. Yes. So these are the different role and different treatment applications of this interferon alpha, beta, and gamma in different disease conditions. So here in this particular question, what they are asking, the primary role of this, like in the case of multiple sclerosis. So in this case of multiple sclerosis, interferon beta 1A is used and not the interferon alpha and the interferon gamma. So that's it. This is the questions of this last week assignment. And these are the topics which are covered till now in until the week 6 material, which is just released recently yesterday itself. So you can check it out the material of this week 6 and also the assignment for week 6 is also released. So you can check that. Yeah, so that's all from my side. If you have any doubt, any question, any suggestion, anything, then most welcome. Are all these questions clear? You understood these questions? Okay. And any doubt with any topic? Okay. Okay. Thank you for attending. If there is any doubt, then you can discuss. You can ask me in this like remaining time. We can discuss that. Or if any question is not clear, then also we can take that again. Yes, yes, those are more than sufficient because the major type of questions are there in the assignments from each and every topic. And in my this live session, I'm also trying to suggest you how to solve the questions. Like you don't just pick out the one option. Like this B is the correct answer for this question. Don't go for this one option. Go for why other five are wrong, why other four are not the correct option. So that in that, if you try to solve the questions in that way, then simultaneously you will learn the five topics in single question. Otherwise, you will see what is the role of which interferon is involved in multiple sclerosis, interferon beta. That's it. Move ahead. Then you will not think about the role of interferon alpha in which disease, gamma in which disease, interleukins in which disease. So you also check out why the other options are wrong. So that is more than sufficient. Okay, and there are I think three live session for this particular course. So you can have you watched other session as well? Yes, that is I think today itself 7 to 9 today only. Yes, yes, on Sunday itself. And one more on Monday. I don't I think 6 to 8 years. One more session is there. So you can see the lecture videos of that also. Like if you don't want to attend the live session of all the this TA session, then you can watch the YouTube videos. Or if you want to attend, you feel like attending, then go for all the three lectures of this three different TA and also the video lecture of Himanshu sir. So everything will be clear in those things. Okay, rest. If you have any doubt, you can post in the discussion forum, or you can drop me an email, or you can ask me in the next class. Okay, happy learning. Okay, so if you want, you can log out, or if you want to discuss, most welcome. Okay, okay, okay. Bye bye. Thank you. Okay, okay.